Good afternoon. A Nevada woman broke her right femur, the big bone in the thigh, while she was in India a couple years back. She had an infection and died because of a superbug, which was resistant to 26 different antibiotics. This news was published on the 12th January 2017. We are currently in the middle of a serious pandemic of antibiotic resistance. What actually is antibiotic resistance? Well, basically, antibiotic resistance is when antibiotic loses its effectiveness towards bacteria. When we're sick, we take antibiotics, but do we actually know what these drugs are for and how do they actually work? Now, antibiotics are very fantastic drugs. They can kill bacteria without even harming human cells. And yes, only bacterial infections, colds and flus, which are caused by viruses, will not be cured by antibiotics. What are bacteria? Bacteria, there are one-celled organisms which does not have a nucleus but has genetic materials. Bacteria, they are here, there, they are literally everywhere. We live in a world full of bacteria. Everything that you touch, everything that you sit on, and even in the bed that you are sleeping in, they are full of bacteria. So, when you thought that you're sleeping alone, actually you're not. In fact, there are actually more bacterial cells than human cells inside you. So basically, right now, technically, I am speaking to a room full of bacteria with some human cell around them. How small are these creatures actually? Well, they are really small that they cannot be seen without a microscope. These small creatures are oftenly associated with diseases, but they are not all bad. In fact, you couldn't survive without some bacteria which is in your body, which helps to digest the food and fight against microbes. So, when we fall sick and take antibiotics, sometimes we not only kill the bad bacteria, but also some of the friendly residential bacteria inside our body. How does antibiotic kill bacteria? Well, the antibiotic and also the bacterium, they work like a lock and a key. Antibiotics, they are designed to be specific in finding and also binding to the targeted site inside the bacterium. However, some antibiotics have shapes which can also fit and also bind with the friendly bacterium and kill them. Though different families of antibiotics, they have different ways of killing bacteria. But all antibiotic does is they inactivate bacteria. So, bacteria, they're amazing organisms in their own way, regardless of either they are bad bacteria or good bacteria. What is it that makes them amazing? Well, bacteria, they are just like us. They love to share. They share genes with each other, and actually, this is how bacteria become resistant to antibiotics. So as you know, we humans, we are made up of genes. Genes are actually what determine what we look like and how we behave. So, for example, bacteria which lives in cold places and can withstand cold temperatures, they will have genes that are resistant to low temperature. And another example is that bacteria which are resistant to antibiotics, for example, penicillin, have genes which are resistant to penicillin. These genes initially arrive by chance through mutation. Our genes are inherited from our parents, right? So, So does bacteria. They can therefore gain resistant gene from their parents, and bacteria can also share genes with each other. How do they actually share these resistant genes with each other? Well, there are several ways on how they can share these resistant genes with other bacteria. One of the ways which bacteria can gain resistance is through picking up genes from their surrounding, and they usually do this when another bacteria dies. Let's say there are two bacteria, bacteria number 1 and bacteria number 2. Bacteria number 1 initially is resistant to antibiotic A. Then, when bacteria number 1 dies off, it will release its resistant genes to the surrounding, and bacterium number 2 will then pick up the resistant genes. Now, bacteria number 2 will be resistant to antibiotic A, and the same thing will be repeated generations after generations, and then a superbug will be formed, which are resistant to all the existing antibiotic. Secondly, virus can also be another mechanism for passing resistance trait between bacteria. The virus will inject bacterium number 1 and package the resistant genes inside the head of the virus. Virus will then attack bacteria number 2 and inject the resistant trait from bacteria number 1. Now, the same thing happens. Bacteria number 2 will now be resistant to antibiotics, which previously only bacterium number 1 can do. Another way of how bacteria can gain antibiotic resistance from another bacteria is by undergoing a simple mating process called conjugation. So basically, in simple terms, bacteria can transfer their genetic material, including genes which encodes for resistance to antibiotics, simply by building a bridge to another bacterium. Antibiotic resistance is inevitable. However, science has enabled a way around this problem. Let's first understand why we take antibiotics. Well, we take antibiotics because we don't want to be ill. What's causing us ill is actually the bacteria. So, we kill bacteria and we are no longer ill. But the problem with this solution is that we develop antibiotic resistance, which ironically can kill us too. What if we can prevent ourselves from getting affected by bacteria without killing it with antibiotics? Now, bacteria, bacteria can only make us sick when there are a lot of them in our body. What happens firstly is that a bacterium gets inside someone, and then the first thing it does is the bacterium divides and grows in number. As it grows, it secretes molecules which can recognize by its own kind. Why do they do this? This is actually their way of communicating with each other. The molecule is as if a message saying, "Hey, I'm here." And how do other bacteria actually read this message? They have proteins, which we call receptor proteins, on the membrane of their cell. These proteins, they have binding sites for the molecules. Again, they fit like a key to a lock. And when there is enough of them, they will start to secrete molecules which can make us sick. This is actually a recent research by a microbiologist named Bonnie L. Basler, and this is called quorum sensing. Now that we know how bacteria communicate with each other, we can stop them. If another molecule can be designed to fit onto the receptors, we will then render the bacteria as useless, because it can no longer bind with molecules which released by bacteria of its kind. Now you have effectively made your bacteria deaf and mute. Can this actually work? The team of scientists infected an animal with a multi-drug resistant bacterium, and at the same time, they injected it with molecules that block the receptor on the bacteria. And the animal lives. We use antibiotics because we want to kill the bad bacteria which cause illness to us, but this leads to antibiotic resistance. Antibiotic resistance is scary and a major problem because most modern medicine rely on the use of antibiotics. To quote Dr. Margaret Chan, the director of World Health Organization, a post-antibiotic era means, in effect, an end to the modern medicine as we know it. Things as common as strep throat or a child's stretched knee could once kill again. Our only solution used to be by making more antibiotics. But as soon as new antibiotics are available, the bacteria began developing ways to resist them. To win in this war against antibiotic resistance, it is essential to invest in advanced researches not only in the evolution of antibiotic-resistant bacteria, but also in all other areas. Science serves its powerful way of understanding how things actually are, and with hope and determination, and with science and its enabling power, an upper hand can be maintained to win the war against rise of the superbugs. Thank you.
We now pass on the Q&A session to the judges.
One question. How do you think the use of antibacterial substances in products such as soaps, hand washes, etc. affect antibiotic resistance in bacteria?
Was there questions about soap, antibacterial soap? Okay.
How do you think antibacterial soaps affect antibiotic resistance? How do they play together?
I think the soap always says that there's 99.9%. It only kills 99.9% of bacteria, right? And actually, I haven't found any proof that the 99.9% actually gets killed off by the soap. So probably the bacteria which didn't get killed off, they probably modify their genes and they get to like resist to the antibacterial soap.
Okay.
Normasturina, right? I get like vice versa. I'm not very. I don't know much about these things, but I do have a question, which is, can you just one more time, just briefly, tell me what is your solution to this?
So there are research that has been going on, and actually it's been proven that it actually works. So in simple terms, researchers have said that when we make a molecule which can bind to the receptor on the bacteria, hence it will like block the binding site of the bacteria. So if we block the receptor, on the receptor on the bacterium. So it can no longer like communicate with the other bacteria. So hence it's like we cut off the communication between two bacteria or more.
Okay. Well, okay. So based on that, I'm just trying to understand. Do you then think that, correct me if I'm wrong, would one of the challenges then be that because everyone is different, do you think that just by creating one, would it be able to apply to everyone? Will it work on everyone? Would bacteria be the same in everyone?
So different bacteria that have different binding sites, the shape of the binding sites. So of course, every one of us are different and we might have different bacteria in our body, either good or bad. But most of the bad bacteria, they actually have the same binding site. So I think it's either the blocking molecule, it does not have to be like one shape. So that's why research has been going on to actually make more of the blocking molecules which can block different types of bacteria.
Thank you.